Today I want to talk a little more about serum amyloid A, or SAA, and whether it really has potential as a biomarker in long COVID. If you remember, last week we examined some data showing that SAA was elevated in blood samples from long COVID patients relative to healthy volunteers, but that the elevated levels were inside the reference range for the test. We also saw that SAA has a role in inflammation, which makes it interesting as a potential biomarker and a good entry point into trying to understand inflammation in long COVID. So I spent this week asking a bunch of questions about SAA. What is SAA's function in inflammation? How is it activated? What happens to blood serum concentrations of SAA during inflammation? What pathways does SAA itself activate once the concentration has increased during the inflammation response? So what are the most important things that I found out? Number one, SAA is what's called an acute phase protein that is induced as part of the, quote, acute phase response, unquote, to environmental insults, including infection, trauma, stress, or tumor growth. The acute phase response is a highly orchestrated and evolutionarily conserved defense mechanism in vertebrates. Once the acute phase response is activated, SAA is synthesized in the liver and then it's released into the bloodstream. Number two, the initiation of the inflammatory acute phase response is predominantly driven by endogenous cytokines IL-1 beta, IL-6, and TNF-alpha. These cytokines induce the expression of acute phase proteins, including SAA and C-reactive protein, or CRP. Number three, high concentrations of SAA activate a number of other receptors and proteins. The two that are most important for inflammation are the FRP2 and TLR2 receptors. SAA binds to both of these receptors on monocytes and initiates more inflammatory and immune response pathways. Number four, activation of the acute phase response by an environmental insult causes a 100-fold to 1,000-fold increase in SAA concentrations in blood plasma. So that means that if the acute phase response and SAA are activated in causing inflammation in long COVID, SAA levels in long COVID patients would be many-fold higher, 10-fold, 100-fold, up to 1,000-fold higher than healthy volunteers. And that's not what was observed in the paper we reviewed last week. So from this, I conclude two things. Number one, the long COVID patients in that study from last week were not experiencing inflammation as a result of the acute phase response. The SAA levels are just not high enough compared to the healthy volunteers. And number two, SAA is not a viable biomarker for long COVID since it's not sufficiently elevated in long COVID patients. Since the acute phase response inflammation pathway is not active in long COVID, the next step is to start examining the chronic inflammation pathways. That's what I'll be working on next. To see a written summary of this info and data, including a lot more information on SAA, check out my bio and click the link. And as always, you can follow my research and give me feedback by leaving a comment below, checking out my bio, and following me. Thanks, and see you next time.